Okay, great. So thanks for having me. So the title of my talk's not really, these are the tools I'll use. I won't talk much about them, but happy to have this opportunity to kind of interface with a group of people that we actually rely on for a lot of the work that we do in terms of the sequencing data that we love to see in terms of the surveillance you guys are doing. So obviously, COVID-19 was an inflection point for society as a whole. Really, it was the equivalent of the 1918 flu to the 20th centuries. This is the 21st century. This is going to shape the next decade, both in term or the next century in terms of infection, our immunity, all of the other aspects. But it's not the first time, and it won't be the last time that infectious disease is going to affect us. You can just catalog this through Time magazines over the last 40, 50 years. Basically, Zika, Ebola, MERS, H1N1, avian influenza, AIDS, polio, flu, herpes viruses. Basically, at every stage in the last 100 years, we've been dealing with infectious disease. The difference is that they accelerate so much quicker because we're a much more connected world. And so in terms of coronaviruses, this was a worldwide, pandemic event that's affected everybody in the world. And the question is, are we ready for the next one? Have we learned? Are we prepared? How can we use what we learned from COVID-19 and apply that forward? And so that's really the goal here. How can we prepare for this next pandemic? So obviously, you guys are doing one thing that I think is important: is surveillance in the zoonotic populations, identifying where these viruses are harbored, where the hotspots that we may or may not know are, where they're coming from. From there, we take it here at UTMB. Obviously, we are one of the premier virology research with the Galveston National Lab, housing both the BSL-4 and BSL-3. There's a huge focus here on emerging viruses. And so, these two elements dovetail with each other. Now, for my group, we use a reverse genetic system. We're really interested in the nitty-gritty molecular biology. And so this is a 7-bit plasmid system that we developed. Originally was developed by Ralph Barrick's group for SARS, MERS, MHV. This is our SARS-2 clone that we published in April of 2020 and that we've been using over the last 3 years. Basically, the system breaks down the coronavirus genome, which is 30 kb, into 7 plasmids. This disrupts the toxic elements and allows amplification of bacteria. We can then sequentially ligate them, drive full-length RNA, and electroporate those into cells and get virus that's equivalent to the epidemic strain. So when we made the WA1 strain in 2020, it had the same characteristics as the clinical isolate that we compared it to. And so in that vein, we're really interested in the basic science and getting down to the nitty-gritty of what the virology is doing. Another element that I won't talk about today is that once we've identified these key tools, these key areas, we want to develop therapeutics and pandemics to prepare for the next outbreak. And this is through an AVID center here. We have an antiviral defense drug development center here at UTMB that we pursue a lot of this work through, but I won't talk about today. And so I stole this figure from one of the Vineet papers that I saw. Basically, surveillance in zoonotic populations we think is really important. There are so many viruses out there, and in 2002, SARS-1 kind of gave us the window for that. And so we know the breadth of the virome is expansive, and you can really see it in the coronavirus family tree. And so here we have the coronavirus tree from 2002, and you can see over the years, they just fill out as more and more animals are surveyed. We've really expanded this family, and this is just a small window into the depth of the viruses that are in the coronavirus family and numerous other families. And so that's one aspect. The other aspect is that geographic diversity means there are viruses everywhere. And so I like to bring these ProMed mail maps. So these are word clouds of infections reported around the world. So this is the report from 2021, broken down by continent and disease location. Obviously, 2021, COVID-19 was really large. I had other ones, but basically these are dynamic. These infections come and go, bubble up, bubble up. You don't know what's going to become the next pandemic, but the key here is that we're constantly infected and constantly exposed to viruses in the population. And so the question is, can we actually predict from a new strain that's surveilled what's going to be the problematic virus or the problematic issue? And so how do we determine risk? What distinguishes an epidemic from a non-epidemic strain? And can we actually identify what will be an emergent strain? I think this is a real question. Surveillance has really populated the virome, but it hasn't told us which strains we need to be worried about. And the reality is, I don't think that if we identify a strain in a bat in Laos or in, Texas, that we could say, oh, that is going to be an epidemic strain. We have to classify it in some way. And what we're looking to do is to build towards that idea. Instead of, looking at one parameter, actually building a list of things that an epidemic strain for coronaviruses needs to have. And so in thinking about that, we've looked at it in terms of the hotspots in the coronavirus genome that allow an identification of risk. So what are these traits? We want to use the number and the composition to then classify risk so that when a new strain is identified, we can quickly categorize, okay, this one has these traits, and these traits are, maybe has 5 out of 6, or maybe has the 3 most important ones, but that'll allow us to classify this compared to other strains that already exist, that we already have in our catalog. And then we can build this framework to select strains for interventions. Obviously, drugs, vaccines, therapeutics, we can't make vaccines and therapeutics for all of them. And so we have to pick and choose. And by generating this classification risk, we can get to that more quickly. And so the question is, what are the hotspots? And so the most common and the major coronavirus hotspot for decades after the coronavirus outbreak was the spike protein. Really, the focus was on the receptor and receptor binding, which is required for entry. And species-specific receptors are required for infection for coronaviruses. So if it doesn't bind to a receptor in that species, it's not a threat. And really, this is the primary barrier for most coronaviruses that we sequence that are in the world. If it can't get into the cell, it's not really a risk. And so a lot of this work was built off of Ralph Barrick's group, where they basically went back to HKU3. This was a SARS-like virus that was the closest known progenitor to SARS-1. And what they found was the barrier for this virus was actually the receptor binding domain. If they swapped in the receptor binding domain from SARS into the full-length HKU3 coronavirus spike, they had a virus that could now replicate in animals. Now, it wasn't equivalent to the epidemic, but it was the major barrier was that this virus couldn't use the ACE2 receptor, the human ACE2 receptor. And so that led to the original paradigm that SARS was actually a random and rare mutation. Random in the sense that it was exposed in a random way, but rare that the vast majority of sequences that existed at the time for coronaviruses really didn't have capacity. And so SARS was thought for the first decade plus to be a rare mutation that allowed it to bind a human receptor. The vast majority of coronaviruses aren't a risk. And so the work that I did in 2015 and when I was in Ralph's lab, we had taken spike proteins from a cluster of bat coronaviruses that were even closer to SARS-1. We put that spike into the epidemic strain and found that those viruses replicated just as well as the epidemic strain. So chimeric viruses expressing the spike from these bat coronaviruses in primary human airway cultures, which is the best in vitro model of human airway disease, those viruses replicated just as well. And so the barrier wasn't quite as large as we originally thought. And so this led to our new paradigm that there are a number of coronaviruses that exist in the population, in zoonotic populations, that have this ability to directly infect humans and potentially cause epidemic disease. But what we know is that while receptor binding is required, is not sufficient to cause epidemic disease. And so in looking at this, we often look at receptor binding as this critical factor. If you can't bind receptor, you can't cause an outbreak or an infection in humans. But we don't actually think that this is what drives epidemic disease. I think there are a number of viruses that have capacity, but they will never become epidemics because they don't have other tools. And so over the last 2-3 years in my lab, we focused on coronavirus entry and spike. And so often the spike is focused on receptor binding. And obviously, for most coronaviruses, to infect a human, they have to bind to a receptor, and this is the major barrier. What's less exposed or evaluated is the second process. So the receptor is like fitting the key into a lock, but the spike processing is actually the turning of the key and opening of the door. This spike processing, we think, can be just as important and just as big a barrier. And so we found viruses in Uganda that are MERS-like, that the barrier for those viruses aren't the ability to bind to a human receptor, it's the ability to use a human protease. And that if you apply protease in trans through trypsin in the media, those viruses are just as viable as the wild type MERS, indicating that this can be just as big a barrier. And so in thinking about receptor binding and proteolytic cleavage, there are two ways that the virus can be cleaved and enter. So there's a cell surface pathway and then an intracellular pathway. And each of these, I think, is important, with the cell surface pathway playing a bigger role in the upper airway and in transmission, and the cell surface or the cell entry pathway more important in the lung and more severe disease. Now, I'll summarize a lot of work that we've done, but we focused early on this region that was downstream below the receptor binding domain in the stalk portion in the C-terminal of S1 at the S1-S2 cleavage site. What we found is that if you take out the furin cleavage site, which increases the efficiency of cleavage of the spike, that attenuates the virus, SARS-CoV-2 specifically. We also found that the loop length is unusual for SARS-CoV-2, that instead of a 13 amino acid coronavirus loop for this cleavage loop, it's actually 17 amino acids in SARS-CoV-2. And so if you reduce that loop length, even if it has a furin cleavage site, it's still attenuated. And so it needed both a combination of a longer loop as well as the furin cleavage site. The third aspect is that there's actually a glycosylation on this loop that's important for protease usage. And so while the virus can be cleaved efficiently, its efficiency in using host proteases at the cell surface is governed by this glycosylation site. And so in case that in this region of the CTS1, the C-terminal of S1 at the cleavage site, that there's a lot of aspects that have to change to drive epidemic disease. These are just three things that we have found. Any one of them reduces the effect, the lethality, the pathogenesis, and the infection capacity. And so all of these elements are things that we would look for going forward. In terms of this, we've actually separated out spike and protease usage as two separate things. And so in making our list, we have this receptor binding domain, and that's a binary result. But in terms of entry, there are several elements that we can think about that drive these differences that we have to go through. But this is not the only place that we're looking. Beyond spike, we've also identified the serine-rich domain of the N protein. And so this region is in a specific region in the linker region of the N protein. It actually shows a mutation in every variant of concern between positions 203 and 205. And what we find is that these mutations actually increase virulence of this virus, increase replication. And so if you take those mutations absent of the other and put it back in the original WA1, we see a significant increase in pathogenesis and lethality. And that actually changes in the context of phosphorylation. The N protein becomes more phosphorylated, which is what we think is driving pathogenesis in the mouse and the hamster and the human models. What's interesting is that doesn't change, and we've started doing work going back to bat cells, and we find that the phosphorylation has shifted, that the original motif from WA1 does better in bat cells than it does in the human cells. And so this may be a molecular switch related to the kinase in bats and humans that are distinguishing and allow this virus to shift. What's interesting is we've actually seen a position 206 in SARS-1, the same mutation, and we currently are characterizing a mutant that is attenuated. So if we put that mutation and revert it back to the bat sequence amino acid, we see it attenuates that virus. Another aspect we're looking at is the 5' UTR. What we found is that if we look at sequencing, and this is work we've done with Andrew Routh, we see conserved 5' UTR mutations in the variants of concern, particularly this U2C that's 100% conserved in the Delta variant, and then 13 U to C at 13C and C21DU, which is conserved in Omicron. What we found is that when we put these mutations back into the infectious clone, into WA1, this attenuates the virus. So we're in the process of evaluating what's the impact of it functionally in terms of pathogenesis as well as transmission. But this may argue that these UTRs may be shifting as a product of the species, and that we can use this as a window into potentially where the virus is in this process of evolving towards something that may become a pandemic. The last aspect to think about is that this is just a handful of sites that we're looking at. There are numerous other possibilities. For example, ORF8 and the accessory ORF are often deleted or cut down in coronavirus infection as they emerge, whereas they're maintained at full length in bat cells or bat virus, bat sequences. And so one of the things we found is that one of our collaborators found that if you delete ORF8, you see an increase in pathogenesis. And we've seen elements of that with the original SARS-1 actually cuts its ORF8 down significantly. And there are some isolates that have been identified that are severely pathogenic that reduce that. And so with all the sequencing data and all the data available to us, we have a number of opportunities to find these hotspots using sequence data as well as surveillance data to kind of compare and contrast what changes as a virus becomes an epidemic. The main takeaway is that surveillance itself is not sufficient. It's critical, but it can't predict the next strain itself. That it has to be paired with basic science approaches to identify these hotspots, and then we can establish this list that builds towards emergence, and emergence risk. Then using this list, we can classify which viruses are a risk or which are the major threats, and then develop therapeutics, whether it be monoclonal antibodies, vaccines, or drugs that target these specific viruses or viruses in this class, so that we have something prepared before the next outbreak, that we can quickly advance and do that. And so with that, I'll just thank my team. Michelle Vu did all of the spike work that you showed. Brian Johnson is leading the N protein work, and then a number of collaborators, including Andrew Routh's group, who's been the primary focus with the 5' UTR stuff. And I'm happy to take any questions.